But before we get too much further into this, I kind of wanted to talk about what I've got set up over here. So basically, to start out this cycle sequencing, it makes it a little bit easier if we've got 4 separate test tubes. In each of the test tubes, we're going to be adding our DNA, which kind of looks like this, but of course, it's in a little double helix. So we're going to be taking a sample of that DNA, the fragment that we want, putting it into each of the test tubes that we have. We're also going to add some polymerase enzymes. We're going to be adding nucleotides, and also dideoxynucleotides. Which are extremely important with what we're trying to do here. So I'm going to explain the difference between the two of those a little bit. So if we had a nitrogenous-based molecule, I'm just going to draw it over here really quickly. It's going to kind of look like this. It's going to have an oxygen and hydrogen group over here, and another hydrogen group coming off the other side. But if we were to have a dideoxynucleotide over here, it's just going to have a hydrogen group and another hydrogen group. There is no oxygen molecule. So basically what that does is when you're going through and you're sequencing your DNA, you're adding nucleotides, what's happening is dehydration synthesis is taking place to fuse that nucleotide with the strand and to sequence and find which nucleotides come next. But since this dideoxynucleotide doesn't have that oxygen, dehydration synthesis can't take place because no water can come out. So basically what that does is it stops the translation of the DNA. And what that ends up doing is it gives us a whole bunch of different fragments of the same DNA that are some are long and some are short. Some can be one nucleotide, others are a little bit longer, a little bit shorter. It keeps going on and on. And what happens is you run this over and over again. It's basically PCR, polymerase chain reaction, just so you can get tons of copies of the DNA. But what ends up happening is, in all likelihood, you're going to end up getting one of these dideoxynucleotides every time that you have a certain nitrogenous base come up. So basically we're dividing it up. If we were starting with A, with adenine, we would run PCR over and over until we have a ton of fragments, and with that comes the increased possibility of having a dideoxynucleotide at least once every time that there is an adenine nucleotide in the sequence. And we do the same thing with guanine and thymine and cytosine until we have all these different sequences with the dideoxynucleotide at least hopefully once every time that it comes up in the sequence. So that's basically what helps us sequence our DNA. I'm going to clear some of this out just so we've got some room to talk a little bit. We've got some room to draw. But going back, we've already taken our DNA and broken it up, so we're working with a single strand instead of the whole double helix. Our primer has bonded. It's telling us where we need to start this whole translation thing. This happens in our test tubes. But once we have all the different fragments of what it could be, let's just label each of the different spaces with a color. Adenene's going to be green. Guanine will be blue. Let's make thymine red. Cytosine can be purple, just to make it easier to see. So we've got all of our different fragments, and what we're going to do is we're going to now sequence it. We're going to run it through a DNA sequencer, which basically kind of looks like a big square. It's a big machine, and it's got a laser down here. I'll show like a laser in red. It's got a laser down here at the bottom. But the fragments of the DNA are going to start up here at the top, and they're going to run their way all the way down to the bottom. And of course, the shorter strands, the ones that have stopped earlier in the DNA cycle, when we were copying our DNA, are going to make it there the fastest. So we run the A, we run adenine, then we run guanine, thymine, and cytosine. And basically, what it tells us is if we had what it comes to compute to, if we had, say that's our DNA sequence, if we're going to start with adenine, it goes through and it tells us that after, say, the first, it was a sequence or a fragment of one nucleotide, and the first one happened to be A. Then the next fragment went through and it was a little bit longer. The next fragment went through and it's a little bit longer, and so on and so forth until you've run out of fragments for adenine. And it tells you in the sequence where each of the adenines are. Then you do the same thing for your guanine, and it says, oh hey look, the third one was guanine, and then a little bit later there's another one, a little bit later there was another guanine and another one, and that runs through. It starts with the shortest and then goes to the longest of the guanine fragments, and that tells us where each of the guanines are in this sequence of DNA. Then we're going to run it with thymine. Shortest one was right there. Next shortest one was over here. Next shortest one was right here. And the longest one was over here. Starting with the shortest, going to the longest, it goes down. And we now know where each of the thymines are in our sequence. And last but not least, we run cytosine, doing the same thing. The shortest was right here. Second shortest came in here. The next shortest comes in there. Next one, and then the longest out here. And basically what this tells us is this gives us each of the different bases and where they are in our fragments. So if we go through and code that, it would be A, T, G, C, A, G, A, C, G, T, no, it's T, got another T, A, B, and C. And so we end up getting at the end is a fully sequenced DNA, so we know where each of the nucleotides are. It's a really cool process, and it definitely helps us with being able to map the genome of different species of viruses, bacteria, animals, humans, anything that has